All right, so today we're going to talk about globin synthesis. So we talked a little bit about heme synthesis yesterday. So make sure you understand that hemoglobin is a protein, okay? Hemoglobin is a protein that is in the the quaternary state. So it has 4 proteins bonded together with those salt bonds. Heme is a part of the globin protein, okay? So heme itself is not a protein, so it's not going to occur or be built at the ribosomes. It's going to be built in the mitochondria and the cytoplasm of the cells. Now the globin synthesis, though, is controlled by chromosomes, all right? So let me see if I can explain this to you guys. So we talked about the dogma of biology is what? DNA, messenger RNA, protein, right? The reason DNA is considered the code of life is because DNA codes for proteins. Does that make sense? You can't live without proteins, and especially enzymes. Every metabolic process that takes place in our body has an enzyme to help it do that, and enzymes are proteins, okay? So that's why we kind of say DNA is the code of life. Because if you don't have DNA, you can't make proteins. If you don't have proteins, you can't live. All right? So when we talk about chromosomes in general, we're talking about genes. Okay? When you talk about chromosomes, you're talking about genes. Genes are nothing but a bunch of what? What are genes? No. What are genes? Well, what are chromosomes? Where are your chromosomes located? DNA, right? So when we talk about chromosomes and genes, we're essentially talking about DNA, right? Okay? So DNA, if we're talking about chromosomes, genes, and DNA, then we're talking about making what? DNA makes what? DNA makes what? Protein.
I said protein.
Right? Well, you didn't say it loud enough. I was being loud.
Yeah.
Globin is a protein. It's a form of protein. So anytime that we're looking at these chromosomes, know that the end result of genes is going to be some type of protein. Okay? Remember, heme didn't have anything to do with the gene, did it? It was done in the mitochondria, right? Chemically made through enzymatic reactions. Okay? Those enzymes were necessary, but they weren't part of the actual protein. Okay? All right, so chromosome 11 and chromosome 16. And this is what we talked about. How many chromosomes does a human have?
46.
Okay? So if we're talking about chromosome 11 and 16, we're talking about 2 chromosomes, correct? Because we have 46, which is interpreted as 2 what? Pairs or sets, right? So how do I have 2 pairs?
Your mom and dad.
That a girl. I have one from mom and one from dad. Because that sperm and egg got together to form you. Okay? Alright, so this is how it works. So these are the ones we're going to look at. So these 2 chromosomes produce a total of 7 types of globin chains. These are going to make Gower 1, Gower 2, Portland. Alright, now these are embryonic. Oops. Alright, then we have hemoglobin F. We have hemoglobin A1, and then we have hemoglobin A2. Now, we also talked about there was like 15 variants of hemoglobin, okay? But these are the main forms that we have. And most of the time, even the variant hemoglobins are still going to be the same as this. Okay? All right, now I have a chart in the book. You actually have the book. It's a little bit further in the PowerPoint, but in your book it's table 6.2, I believe. What's that table right there? Yeah, 6.2 on page, what is that, Kara?
80.
Page 80 of your book. There is a table that gives you all of these listed, okay, and then it gives you percentages of these, okay? So, but right now, this is what we want to look at, all right? So, alpha 2 and gamma 2, alpha 2 and, what's that delta? Is that the delta symbol? Kind of like that? Close enough?
Yep.
Alpha 2 and delta 2, okay? All right? So, these two chromosomes play a role in helping us make these hemoglobins. These two, we're going to talk about these in just a minute, but right now, just this is important. These two or three, right there. Okay, you need to know those three. All right.
The bottom three.
Yeah, these are what you need to know. Okay. All right. Here's why, guys. Because most of the time, 99.99999% of the time, we're never going to do embryonic hemoglobin, right? And that's what these guys are. So we don't really care about these, okay? We're going to talk about them just so you'll know what's going on, but we're never going to see this in a lab. Does that make sense? Okay? These we will, okay? Because every red blood cell in your body has hemoglobin A1 about 95 to 97% inside. Hemoglobin F is about 2%, okay? And then hemoglobin A2 is about 2%. It's actually about 1 to 2, depending on what you got.
Is
okay?
this, I'm sorry, is
That's
this
all right.
in your notes somewhere?
Yeah, eventually. It's in that, on that chart. Look right here. Look right here. See those percentages? Okay. All right, so every red cell in our body has these hemoglobins in it, these three, okay? Even though I'm not a fetus anymore, I still have a little bit of fetal hemoglobin in my blood, okay? So if I could suck out the hemoglobin of one red cell, if I am a normal human and don't have a hemoglobin variant, I should have 95 to 97% hemoglobin A1. A lot of times it's just referred to as A, okay? I should have about 2% of hemoglobin F, which is fetal hemoglobin, okay? And then I should have about 1 to 2% of hemoglobin A2, all right? Okay? So if I look at the composition, hemoglobin F is made up of alpha and gamma, okay? If I look at A1, which is my normal majority of my hemoglobin, alpha 2, beta 2. Okay? And A2 is 2 alpha, 2 delta. Right?
Is it always the letter before the remainder?
Say it again.
So I put 2A on my notes. That's wrong. That'll
A2 is what it's going to look like. This is what it's going to look like. I mean, I don't care what you put, but this is what it's going to look like. Okay. All right. Questions about that right this second?
What's the first one, the 97%? The AG?
A1, hemoglobin A1, this guy. Second one's F, and then the other one's A2. Okay. You can make your red cell bigger than I made mine if you want to. Or you don't even have to put it inside a red cell. It doesn't really matter. I just did it because that's what I did.
Because that's what
All right. So, if you remember, we have 7 types of globulin. Okay, so you really probably think there's only, what, 1, 2, 3, 4, 5, 6 types. Okay, the difference is gamma. The gamma has 2 different forms. Okay, it has a gamma A and a gamma G. All right, to us, it doesn't matter. Just know that it's a gamma, okay, which is this guy, okay? Fetal hemoglobin is made by gamma, okay? All right, so the genes for the zeta and the alpha are located on chromosome 16, okay? So you have a loci that looks like this, okay? You have your zeta and your zeta from mom and dad. Then you have an alpha 1 and an alpha 2 from mom and dad. All right? So, we're going to be really, really logical because that's what we do as scientists, right? And whether you wanted to be a scientist or not, you are a scientist, okay? At this point, you're wanting to be a scientist, okay? So, we're going to use logic all the time. All right? So, if I tell you that all the rest of these epsilon in it. Epsilon. So you got two O's, is that right?
It's
EPSI. Okay, thank you. And I'm just going to put G and B and D on this one, okay, because you know they won't work the same way. All right? So, these guys are on chromosome 11, okay? But what's unique about these compared to these?
these require more amino acids.
Nope.
What?
Well, it may be true, but that's not what I'm looking for. Okay? What's unique about alpha compared to all the other ones over there? That's right. There's two alphas on each chromosome. Whereas over here, we've got two of each, but in total, how many we have over here? We have 4. Why do you think that's important? Why would it be important? That a girl. Because it's in every one of them. And if you look on page, what is that, 80 you said? Look at my, tell me the Gower. Gower 1 and Gower 2. Doesn't it have the composition of it also?
Not on that.
Yeah,
Yeah, right here.
Gower 1 is zeta 2.
Ah, correct. All right, so that's that little weird, like that, right? Zeta 2 and then what, babe?
Epsilon 2.
So E. Okay. All right, and then the Gower 2 is what?
Alpha 2.
And then what's Portland? Back Zeta, isn't it?
Zeta 2, gamma 2.
Okay. All right. So, all right. So this is the time. Look, this one starts about 1 to 2 weeks of gestation. So this guy is actually formed where?
The yolk sac.
The yolk sac, right? Because remember, that's where the blood really starts forming in hematopoiesis, is in the yolk sac, okay? So these don't last very long. And then look what takes over. Alpha, okay? Now, it does revert back to zeta, okay? But then look, once we get at a certain point, and actually, I think it's at about 30 weeks, it may be a little bit earlier. At about 30 weeks, we convert to this, and we start making fetal hemoglobin. So what happens, okay, so have you ever heard this term? Two terms. You ever heard the two terms upregulate and downregulate? Okay, so you remember the other day, a couple of weeks ago, we made, we were talking about the genetics and all those kind of things, and we used the example, I believe it was J-Lo, that was positive for the two. Did we have this conversation? Must have been with the other class when we were doing clinical chemistry. Okay, so when you have genes, okay, we know the human genome now. You know, we got the human genome 1998, I believe it was. We basically coded every gene in the human body, okay? Now, we didn't know exactly what those genes were, but we knew that they were there, okay? So now, 2022, we know a lot of those genes. And we know that there is a, and help me, what is it, BCR? BRCA, yeah. All right, we know that if you get this gene, you have an increase in developing breast cancer, okay? So just like every gene that we have in our body, no matter what, how many copies are we going to have? We're going to have 2 of them. So we know that if a person ends up being homozygous for that gene, and they have it times 2, 1 on each chromosome, their chance of going up is like 80% of developing breast cancer. Right? I know that. Okay? All right, well, if you didn't know that, you do now. Okay, very good. So, J-Lo, who's a movie star, and, you know, is worried about her looks and everything, like a movie star should, I guess, she tested positive for both genes. So instead of waiting till she developed breast cancer, she had a double mastectomy and reconstructive surgery and got her some store-bought boobs, right? Okay. Was that smart?
Yes.
Does it mean she's going to get breast cancer?
Well, she's got the money to.
But is she going to get breast cancer?
Probably.
Is she going to get breast cancer?
Yeah.
80% chance that she.
Is she going to get breast cancer?
We don't know.
We don't know. You know why we don't know? Is because just because you have a gene, there's still a 20% chance she won't. What does that mean? That means that if that gene gets upregulated and turned on, yes, she's probably going to fall into that 80% category. But what if her body suppresses and downregulates that gene? Is she going to get breast cancer?
20%.
Probably not. 20% chance that she's probably not going to get that gene or get breast cancer. Does that make sense? Okay? So, that's what I want to talk about here. So, the way our genes work in our body, remember how we talked about efficiency our body is? Okay? At a certain point in our time, in our production or our growth, we don't need this guy anymore. Does that make sense? So what happens, our body sends a chemical signal to these 2 genes to say, look, dudes, y'all need to cut yourself off. We don't need you anymore. That's called downregulate. Okay? So it does. So then when that happens, though, what's got to happen to these 2 genes? They have to upregulate, because now our body says, well, we got to have hemoglobin because we kind of like to breathe. You know, we like oxygen, and that's the only way we can transport it. It says you guys need to kick in because we're going to need you to make this, and we're also going to need you to make that for a little while. Okay? So you get a promotion. So you start kicking out those alpha globins. Okay? So the gene, the messenger RNA to make alpha globins is going to go and go to those ribosomes, and we're going to start churning it out. Okay? Now, the reason that there are 4 of these is because it is so important for us as adults, right? Because every hemoglobin we have from the time we're 30 weeks gestation has to have those alpha genes in it. If we don't, we can't survive because we can't make the right hemoglobin, okay? It's technically not true, but it's harder for us to make hemoglobin. And we'll talk about that when we start talking about hemoglobinopathies and thalassemias, okay? All right? But does that make sense to you guys? So these guys, though, we only need 2 copies of them because eventually, what's going to happen to this guy? Epsilon. What's going to happen to it? Do you see it anywhere over here? No, he's only needed right here, right? For these 2, actually. So very early in gestation. So after that, what's going to happen? Body's going to say, hey, downregulate. We don't need you anymore, right? Then he's going to kick up production of these, okay? And this one, right now, this one is probably up more because, look, we need to have fetal hemoglobin, which is what we need in the womb, okay? And then for a little while after we're born, we still keep fetal hemoglobin, okay? But then after that, what's going to happen to this guy? He's going to get downgraded a little bit, okay? And then these 2 need to kick in to production. And then mostly this guy. This is the one we want to upregulate mostly, okay? A lot, all right? As we go, because that's what we need. We need 97% of our hemoglobin needs to be 2 alpha and 2 beta. Okay? That makes sense to you guys? Okay, excellent. All right, so group chromosome 16, chromosome 11, 2 very important chromosomes, and keep these 2 in mind, okay? We're going to need these guys again when we get into thalassemias, okay? All right, so keep those in your mind. Chromosome 11, chromosome 16, know which ones they code for, all right, which genes are on which one, all right? Because we're going to see it again, I promise, guys, see it again, okay? All right, so zeta synthesized very early in that embryonic development of the Gower-1 into Portland. Then zeta is replaced by alpha, so zeta gets downregulated. Alpha gets upregulated about 8 to 12 weeks, okay? So notice, like we was talking about, there are 2 loci, and that's the term you use for a gene location, okay? If you're just talking about one, it'd just be location, but when you talk about both chromosomes, then they refer to it as a loci, okay? Two spots on the gene. Both of these transcribe messenger RNA for structurally identical alpha chain, okay? So what that means is, just because we have alpha 1 and alpha 2 doesn't mean we have 2 different forms of alpha, okay? They're both genetically to make the same globin chain. We just got 4 chances to do it, okay? And that's going to come into play when we start talking about the thalassemias. All right? Then you've got the chromosome 11, which has your non-alpha ones. So if you guys learn the alpha, alpha goes with 16, non-alpha goes with 11. Okay? Does that make sense? So you had it on your quiz today, the alpha and the non-alphas. Okay? So now you just need to add which chromosome's which. Okay? 16 is the alphas because it has 4 alphas on it, you know. And then chromosome 11 has the non-alphas. Okay? And this is what we was talking about here with the switching on, switching off. That means upregulate, downregulate. All right, so we're not talking much about that because I've already done a pretty good job of that. Okay? What did I miss?
Figure out where you're at.
I am on slide 122.
She should.
Okay. All right. So non-alpha globin genes are found on the chromosome 11. That's delta and beta chains, which are the 2 that are activated the most for us. And therefore, then they're going to be, the delta will be downregulated and the beta will be upregulated because hemoglobin A, listen to me, the major adult hemoglobin is hemoglobin A, or hemoglobin A1. It's the same thing, okay? We put 1 when we're comparing it to 2 down there, all right? Some books leave off the 1, but just know that this is hemoglobin A1. Okay, all right, and then it makes up 97% of your cells. So if I ask you what's the composition of the major adult hemoglobin, what would you tell me?
Alpha 2, beta 2.
2 alpha, 2 beta,
Can you
right?
ask the question again?
What is the composition of the major adult hemoglobin? Okay, 2 alpha, 2 beta. Very important. Okay? Make sure you know that. All right. Questions about that, guys? All right. Now, these guys do occur, of course, on ribosomes because we're making protein, right? So this is where they occur. All right. They create a hydrophobic pocket near the exterior surface of the chain, and then the heme molecule is then inserted into the pocket and is readily accessible to oxygen. Okay? All right. And of course, remember, this is always occurring where? In the liver. No. Well, let me ask you this. Globin chains are made to do what? Create what? Hemoglobin. Where did we say the only place hemoglobin is made in our body? What's the big term for all of them? Because it's made from the beginning one, the erythroblast. Right? So in other words, the nucleated red cells up to the metarube. Okay? Metarubes don't make it anymore. They're already complete, about 80% of it by the time it gets to the metarube. Okay?
So this is happening.
So this is happening in the erythroblast. Okay? Guess what? It's the only place it's happening. Okay? Because that's the only place it's necessary. Right? No other cell in our body, well, shouldn't really say that. The 99.8% of the hemoglobin in our body is made in which cells? Erythroblasts. Okay? Because it's actually a little bit crazy. A couple of the white cells have a little hemoglobin in them. Okay? It functions just like regular hemoglobin, but we just don't consider it because it's not the major source. Okay? I know that sounds crazy, but all right. So hemoglobin. So this is all occurring at the ribosomes of the erythroblast. Okay? All right. Trying to see if we've talked about this. Okay. So basically, you start out as a dimer. Okay. Anytime you see the word dimer, okay, 2 dimers combine to form a tetrameric hemoglobin molecule. Okay. This is a dimer. That's a dimer. Okay? 1 alpha, 1 beta is a dimer. Okay? So then it binds with another beta and another alpha. Okay? And then that's 2 dimers. 2 dimers. Okay? Then another alpha and beta form. Okay? To form the tetra. They form a bond here, and a bond here, and a bond here. Okay? So remember I drew out the little squiggly things the other day? Yes, or Monday? Right? Remember that? Okay? That's the same as that.
And they pick up oxygen.
Correct. That's exactly right. So they would have that little heme right in the center of them, in that little pocket, and then that's where your O2 would form with iron. Okay, because remember, heme has iron in it. And iron in what form? Ferrous, which is F2. Fe plus 2. Tetrameric is what I really wanted to put. I may have put tetrad, but that would be the same thing. Okay. Four of them go together. Okay, so 2 dimers form affinity. Okay, are you married? Do you like your husband? You don't like your husband? Do you love your husband at least?
Yes.
So you have an affinity to him. In other words, you had an attraction to him. Okay? Now there's a difference between, we're going to talk about it in just a second, we're going to talk about affinity and avidity. Okay? They're 2 different things. Avidity. Affinity is an attraction. Okay? I'm attracted to my wife. I love her. I've loved her since we were 15. We've been together since we were high school sweethearts. Okay? We've been married 23 24. Be 24 years this year in April. So I have an affinity for her. Okay. She also is a sugar mama. She makes 6 figures a year. You know what I mean? So I also have an avidity to her. I want to do whatever it takes to hold on to her. See the difference? So affinity is an attraction, and avidity is the ability to hold on to it. Okay? And that's important. Those two terms are really, really important. Should have learned them in chemistry, but if you didn't, we're going to learn them today. Because we're actually going to talk about something that has to do with this in just a second. Okay? Questions about anything?
How do you spell the second word you're saying?
Avidity. I don't know. I have to write it down. So affinity, AFFINITY. Avidity. No, that doesn't look right. No, is that right? You got an extra I? Avidity. I think it's pretty close. I may be a little bit off, but it's close. It's in the book. We're going to talk about it in just a minute. Okay? All right. So we have this protein. It's known as the alpha hemoglobin stabilizing protein. And basically, its main role is just basically coordinating the assembly of heme and globin together, getting them together. So if you don't have this protein, what might cause you a problem? What would the problem be? If you're missing AHSP, alpha hemoglobin stabilizing protein, what disorder might you develop?
Iron deficiency?
Which is a form of what? Anemia. There you go. You're probably going to develop an anemia for this because you're not making hemoglobin, so therefore your body's not going to make what?
Well, wait, what'd you say?
Yep, if your body's not going to make hemoglobin, what's the use of making what? Say it.
Blood.
What kind of blood? Remember, we're scientists. We can't just say blood anymore. Red blood cells. That's exactly right. And what's the definition of anemia? A reduction in RBC production. Okay? So make sure you know that. So that's one of the things that can happen if we're missing this protein. Of course, it'd be genetic and that kind of thing because you're missing the protein. Okay? All right, so this is what hemoglobin looks like. Your little dimers getting together to make a tetramer. And then this is the heme structure itself, okay? So this is what was formed in the mitochondria, okay? And then you notice that the iron is inserted right in the center. Notice the iron is between four nitrogen molecules, okay? Those nitrogen molecules act very similar to oxygen. Okay, so it has some helps with the affinity and avidity of the. I'll tell you what I messed up over there. Now that I'm thinking about it, avidity. I didn't have hooked on phonics when I was a kid. Avidity. That's exactly what it is. That's the word. Avidity. I didn't have hooked on phonics. Sorry. Not before, that was before my time, or after my time, okay? So balance synthesis is very important between heme and globin, okay? So every time you break down hemoglobin, you make hemoglobin, okay? Most of the time, though, globin can be recycled, whereas heme gets broken down. And we're going to talk about that in just a few minutes, okay? As we go, all right? Synthesis, of course, we talked a little bit about this. The regulation has to do with that ALAS1, which is what? Or 2, I'm sorry. What is this? What did I say this was?
An enzyme?
That enzyme does what?
It makes an acid, like the ALA.
All right, where did this right here come from? I mean, yeah, it's the 5-ALA, right? Where did that come from? Do you remember?
Is that not from the succinyl?
There you go. It's from the succinyl and the glycine. So it takes for them to bond chemically to form a new compound, an enzyme has to be placed into it. And that enzyme is this. So this is a limiting step. If we don't have this particular enzyme, you can't make what you have. Even though you may have plenty of succinyl-CoA and plenty of glycine, if you don't have this step, you can't regulate or you can't make hemoglobin. Okay, so it's a regulator. All right, without these there, you basically get messed up. And then the concentration of iron also plays a role in regulation. If we don't have any iron, our body says, well, we don't have iron, why are we making hemoglobin? Iron is the only thing that can bind to oxygen. So, therefore, no reason to make hemoglobin. Okay? All right? So, iron is very, very important. The pool available for metabolic processes, heme synthesis, the storage of pool, is we'll talk about ferritin and hemosiderin. Ferritin is the storage form of iron. Okay? Hemosiderin also is too. Hemosiderin is usually when it's in the bone marrow. We call it hemosiderin. When it's in the what? Storage and metabolism. The answer is always what?
Liver.
Liver. When it's in the liver, we really consider it ferritin. Okay? So, but ferritin is the form of storage iron. And we'll talk more about that later when we get into IDAs. But you can go ahead and see those words. Okay? All right. So iron concentration is very, very important to us for this. All right. Ontogeny of hemoglobin is basically how it's the genes and those kind of things that play a role in producing it. So we've already kind of talked about this, so you guys can kind of read through this. It talks about up-regulating and down-regulating, which it talks about switching. When it talks about switching, when it switches, it either up-regulates or down-regulates, is essentially what it's talking about. Okay. And then, of course, up to a year, after a year, predominantly it's going to be hemoglobin A. Less than a year, it's going to be predominantly hemoglobin F. Okay, so instead of us having 2% of fetal hemoglobin, if I draw blood on a 2-month-old and do electrophoresis, well, that's a new term. We'll talk about hemoglobin electrophoresis a little later on, but anyway, it won't be on your test, so don't worry about it, okay? Hemoglobin electrophoresis, instead of getting 97% of hemoglobin A1, 2% fetal, 1 to 2% of the A2, I would get probably 90%, 95% fetal hemoglobin, hemoglobin F, and then a little bit of A and a little bit of A2, okay? But then after a year old, I should revert back to making adult hemoglobin. Okay, so in other words, that beta gene gets upregulated, delta and gamma get knocked down, downregulated. Okay, here's this little chart. You guys can look at this. So let's from fetal to a year old. This is the what you're going to see. So 90 to 95% is what they're calling, and then after birth, 50 to 85% instead of the 95%. Okay. So they're still making some A here, even at between a year, less than a year old and being born, okay? They're still making, and they actually, let's see if I can explain this to you without being too confusing. So like, once they get to about 30 weeks, and that this zeta gets knocked back down regulated, and alpha kicks in, we start producing this. Alpha and beta also get kicked on, okay? They're just not being, they're being down-regulated, but they're producing. Does that make sense? So you're not going to see a lot of percentage, but they are making it. In other words, they would be detectable amounts if you tested for them. Okay? But then once they reach a year age, a little over a year, then their body says, okay, gamma, we can cut back, and then beta, you kick in, okay?
HbA.
Huh?
And that does HbA.
Yes, A1. Okay. All right, so talk about fetal hemoglobin for just a minute. Fetal hemoglobin is a unique hemoglobin. It's predominant during the liver and the bone marrow, early bone marrow. So remember, after the yolk sac gets deregulated or downplayed, the liver forms, then we have some bone marrow formation, okay? So then those cells start forming hemoglobin F, okay? 90 to 95% until about 34 to 36 weeks, okay? So I put 30, but you know, I was just going by the top of my head, okay? Till gestation, and then when you're born, it drops back down to 50 to 85%. 2 alpha, 2 gamma. Need to know that. 2 alpha, 2 gamma, okay? Fetal hemoglobin is a unique hemoglobin, so we'll go ahead and throw some extra in for you guys. We do I always have to look up this this name. Somebody Google fetal stain for me. Somebody got Google. Hell, y'all got Google. Tell me how, does it pop up Kleinhauer Becky? Spell it for me. Spell Kleinhauer for me.
Kleinhauer.
Okay, hang on. Hauer. And then the last one is, it's like B-E-K.
Yeah,
T.
like that.
K. K-E. Okay? This is a stain that you guys will learn about in the blood bank when you get a little further, but I am going to go ahead and mention it to you because we're talking about fetal hemoglobin. So this one is used, this is a stain. So essentially what you do, it's a German guy, you guys didn't figure it out. The German people were smart. Back in the day, when a lot of the things were being discovered and that kind of stuff, a lot of it was German scientists, because Germany saw the need to put money into research, scientific research, more than we did in America, okay? So basically what happens, guys, is you put a drop of mother's blood, okay, so draw a purple tube from mom, is essentially what you do, a lavender tube, and then you make a smear with her blood, okay? Then we're going to add acid, wash, okay? It's usually hydrochloric acid, maybe phosphoric acid, just depends on which company and which brand, okay? But it's basically an acid wash. Here's the deal. Fetal hemoglobin, so hemoglobin F is resistant to acid, okay? So what does that mean? It means that all the little fetal blood cells from the baby that are on the slide are going to stay intact, okay? Mama's blood, which is going to be full of what? Hemoglobin A, is non-resistant. So what's going to happen to her cells? They're gonna lyse, okay? So mom's cells all die, or they basically get washed out, the hemoglobin. And it's really funny because you'll still see the red cells, you'll just see the membrane is all you'll see. We call them ghost cells, because they have no hemoglobin in them. It's literally just like a balloon after you busted the water out of it, of a water balloon, okay? That's exactly what it is, okay? So then you stain it with Kleinhauer-Becky stain, and these guys, it's going to pick up the stain. And so you get these bright red cells that are stained, and those are fetal cells, okay? So why do we do this? We do this to calculate ROGAM. You know what ROGAM is? Y'all ever heard that term? How many of y'all are RH negative? Any young girls RH negative? No? Y'all are all positive? Okay. Well, you'll find out in the summer. Y'all guys will do your own blood types this summer. That's part of your immunology course. You guys will do your own blood types and stuff, so you'll know you'll actually go down, and when you get into blood bank, you guys will even do antigen typing. Y'all know exactly what your RH types are C, big E, little E, big C, those kind of things. Y'all know all that stuff. Duffy, Kid, all that. She'll let y'all do all of it. Rogam, okay? All right. I mean, history people. Man, how many of you like history? None of you? So that's what's wrong. That's what's wrong with America right now. Go ahead and be political. It's because y'all don't know history. Because you had PE coaches for history. I love history. Yeah, I love history. Okay? History is something you need to learn so you don't repeat it. We don't need to cancel it out like we're doing today. Okay? History is important because you learn from it. You need to learn not to do that again. You know? One thing history people do is they like to visit cemeteries. And I know that sounds crazy, but you get to see lots of ages, lots of years, people, those kind of things. Okay? How many of you have been in cemeteries where you see this? You see like mom, and then you see all these little baby ones. And usually they'll be anywhere from like a day old up to about 7 days old. And then they die. Okay? And usually it's multiple ones. Well, research showed us or told us as we went through, they did enough research to find that usually there was a kid right here between this one. Okay? This mom was found to be Rh negative. Okay? So what happens is this baby was probably Rh positive. So when she had Junior, okay, if y'all have ever seen that little situation,
Oh my goodness.
when Junior comes out, it's bloody, nasty. Like St. Bernard coming through the cat door kind of thing. You know what I mean? Just gross, right? So what happens is Junior's positive, mom's negative. Blood gets into mom's bloodstream. Okay? Junior's blood gets into mom's bloodstream. So now she's got little positive red cells floating around in her body. What's her body think?
An invader.
It's an invader. And what do we do when we have an invader?
Figure.
Our B cells do what? They become activated. And then they make a what cell? And then that plasma cell makes antibodies. Okay? And in this case, it is anti-D, which is what makes our blood positive. It's a protein on our red cells. It's called a D antigen. Okay? The problem is, it's this. IgG. And IgG is a mean antibody. Okay? It's the one we want to make all the time. Anytime we make an antibody, we usually start out as an IgM, but we want it to convert to IgG because it's a mean dude. Okay? Another thing about it is it's really, really tiny. And guess what it can do? Cross the placenta. Okay? So, this Junior is fine. He's normal, he's good, everything goes good with him. Okay? But this Junior is also Rh positive. So when mom gets pregnant with Junior again, this Junior, who's Rh positive, her body says what? He's foreign. Because those are that's positive blood and we're negative. So her body makes an antibody that crosses the placenta and Junior dies. Okay? A lot of times if you do the research, they also had several miscarriages before they were able to have a baby. And then this baby didn't do good because we get fetal hemolytic disease of the newborn, is what it's called. And so her antibody attached to all of his cells in his body once he did become born. And so it popped all of his cells. And he gets jaundice and dies, essentially what happens. So, all that to say this. So this all occurred before 1940s. So I don't remember, maybe like 47 or whatever. 1947, guy figured out what was going on and he said, well, hey, why don't we make a synthetic, so how do you spell synthetic? That's not right, it's got an E right there. Synthetic anti-D, it doesn't? Okay, so I was right then? Okay. Synthetic anti-D, okay? So what he did was he said, well, we'll shoot them up at a certain point in their pregnancy, and it's actually 28 weeks is what they do it at, okay? Then if mom's negative, we give her the anti-D to start with. Then once baby's born, if she has any of that baby blood in her, these antibodies will attach to it before her brain and body can say, hey, we got a foreign invader. And it goes ahead and kills them. So she never makes natural anti-D. And so now the next baby's okay. Problem is, and the reason we do this is because it is really, really bloody. And so we don't know how much RhoGAM she's going to need afterwards. And so we count the number of fetal cells based off the number of her cells, because you can still see the membranes, okay? And then we do a calculation. So she may get a couple of shots to be able to keep her from forming natural D. Yes? RhoGAM.
Is this whole process?
That's it. Well, the process is why we use RhoGAM.
Okay, RhoGAM is the synthetic.
Anti-D. And then these guys, this is the stain that we use. It's called a Klein-Hauer-Becky. Okay? So fetal hemoglobin is a unique protein because it is resistant to acid. So we can use it for lots of things. They can actually use this thing too, and you may see this sometime in the lab when you're working, is mom's in a car wreck and they want you to do a Klein-Hauer-Becky on them. Okay? What they want to do is see if baby got hurt and see how much bleeding baby's got. You know? And so it may not have anything to do with RhoGAM, okay? So you guys need to know that too. The Klein-Hauer-Becky is to detect fetal hemoglobin, is what it is, okay? All right, all right. So adult hemoglobin. So we know hemoglobin A is the predominant major hemoglobin. Formed as early as 9 weeks gestation, but not enough of it to really do any good. So that's the reason we have that Gower 1, Gower 2 in Portland. And then eventually, though, it does kick in. So no alpha 2, beta 2 is hemoglobin A. Hemoglobin A1 is the same thing. Okay. All right, about 95% at 1 year of age. We talked about that. Then as soon as adult hemoglobin is produced, hemoglobin F should decrease. We're going to talk about thalassemias. Beta thalassemia, that's one of the characteristics of beta. People that have beta thalassemia is they actually have an increase in hemoglobin F in their blood, and that's an indicator that they have it, okay? We talked about all this. Hemoglobin A2 is very little. It does start occurring late in fetal life, but less than 1% of adult hemoglobin. Normal adult values say 1.5 to 3.7. I think I put 1 to 2 on the board, so it all just depends on where you're at. Actually has a higher affinity for oxygen than A2 does. I mean A1, which is kind of crazy, but fetal hemoglobin actually does too. Fetal hemoglobin actually has a higher affinity and avidity for oxygen than hemoglobin A does. Okay, but I've already told you about affinity and avidity. It's okay to have a high affinity, but do we really want the protein to have a high avidity for oxygen? No, because what do we want oxygen to do? We want it to eventually do what? Go out into the tissue, right? And if the protein has a high avidity for it, it doesn't let it go. It wants to hold on to it. Does that make sense? So even though hemoglobin A2 and hemoglobin F have high affinity for oxygen, they also have a high avidity. And so that causes us a problem, okay? All right, let's talk about glycosylated, all right? Glycosylated hemoglobin, this one's important to you guys. Hemoglobin A1, which is the major hemoglobin, but glycosylated hemoglobin is broken down into 3 other ones. Hemoglobin A1, hemoglobin A1A, hemoglobin A1B, and then hemoglobin, the one that we're concerned with, hemoglobin A1C. Okay? This is the guy that we're concerned with. All right. So what does that mean? All right. So what it means is, what does glycosylate mean? You leaving me?
I have to go get my kid.
We'll go get your kid.
Our normal teacher's not there today, and she stays later for me, so I can get
okay.
there after class, which has a different teacher today,
That's
so
all right.
I can't be late.
You got it.
But
It's
I'll
all
finish
good.
watching it
Listen to the
on
lecture.
YouTube.
Okay?
I will.
So glycosylate. What's glycosylate mean? What's glyco?
Sugar.
Sugar. What? No, it's not sugar.
Glucose.
We're glucose, right? Remember, we're scientists now. Okay. Glucose. All right. So glycosylated means basically exposed to glucose. Okay. So what happens is, the more a substance or the hemoglobin is exposed to glucose, the more it tries to pull glucose into it. Okay. So it's called glycosylated hemoglobin. Okay. So, all right. Any of y'all guys diabetics? You're diabetic, type 2, I'm sure, right? Because I don't see a pump and I don't, you know, so I'm going to assume. Okay, so did y'all ever meet Brian Lowe? He's in our other class. Curly headed, wears glasses, real quiet, doesn't ever say a whole lot. Younger guy?
I think I know it because it's on the bookstore, but I
Yeah,
don't know.
yeah. Well, Brian is a diabetic. He's a type 1 diabetic. So like he was like 6, I think, or 5 when he got diagnosed with having diabetes. Okay? So, people who are diabetics, you don't go to the doctor, which they do, but they really don't care if you come in fasting and your glucose is normal. Okay? They don't care about that anymore. They really don't. But what they do want to know is your hemoglobin A1C. Why? Because if I'm a diabetic, okay, and I know my doctor's appointment's in 2 weeks, and I want to get a good report for my glucose level, all I have to do is don't eat any sweets like a week before I go. Matter of fact, about 3 days before I go, and I can keep my glucose level down, okay? But as soon as the doctor does my hemoglobin A1C, he's going to know that I stayed, like, work part-time at the Krispy Kreme. You know what I mean? Like,
There's no hiding.
you know, there's no hiding, okay? Because what happens is, all right, so when our plasma glucose level goes above 400, glycosylation occurs, okay? So what that means is these 3 types of hemoglobin of A1 will pick up glucose because there's so much glucose in our plasma, our body don't know where to put it, okay? It tries to store it in our cells, it tries to break it down into glycogen to store in the liver, but there's just so much of it, we can't. And so it says, okay, hemoglobin, you got to take some, you got to take some, you got to take some, okay? So when that glycolated hemoglobin or glycosylated hemoglobin level goes up, my doctor knows that my blood glucose has stayed above 400 for a while, okay? So we call it the cheater, okay? If you're a cheater diabetic, your hemoglobin A1C will tell on you, okay? Does that make sense? No?
Is it 400 milligrams
Per deciliter. Yeah. So hemoglobin A1C, we like for it to be between 3 and 5%. Okay? That's normal. Okay? Because listen, even if I'm not a diabetic and I go to the Krispy Kreme and I eat a dozen donuts, because you need them hot like that, you know, you need a dozen before you know it, right? What's my blood sugar going to do? It's going to go up. It can't help it. Even though I'm normal and don't have any kind of diabetes or anything like that, my blood sugar is still going to go up. My glucose, see, I did it too. My blood glucose is going to go up because it's just the way my body works, okay? So we're going to be allowed to have a little bit of hemoglobin, okay? But when it gets up to like 15%, what's Doc know? Doc knows you've been living at the Krispy Kreme. You know what I mean? You're like back there, like pouring sugar down your throat, you know, that kind of thing. So, you know, so diabetics, if you're a true diabetic, they want it to fall between that 5 and 7%. is what they like for diabetics to stay. Okay? All right, questions about hemoglobin A1C. That's the important one. I know there's 3 of them, but A1C is the one that we're concerned with in the lab. All right, talked about that. See, they said 7.5. Sorry, 3.5 to 5 is what the range actually is. Let's see. All right, questions about that? Okay, so today's Wednesday, so we're not going to finish because